Today, an FDA approval in tenosynovial giant cell tumor, a breakthrough therapy designation in melanoma, and encouraging findings in trials in bladder cancer, lung cancer, and a cell-free DNA assay. Welcome to OncLive News Network. I'm Kristi Rosa. The FDA has approved pexidartinib, known by the trade name Turalio, for the treatment of adult patients with symptomatic tenosynovial giant cell tumor that is associated with severe morbidity or functional limitations and not responsive to improvement with surgery. The approval of the small molecule CSF1R receptor inhibitor was based on findings from the Phase 3 ENLIVEN study, which demonstrated a 38% overall response rate with pexidartinib compared with 0% with placebo, following 25 weeks of treatment based on central review of MRI scans. Additional data showed that the complete response rate was 15% and the partial response rate was 23%. The median duration of response was not reached with pexidartinib and not available with placebo. 96% of patients had a DOR of at least 6 months, and 50% of patients had a DOR of at least 12 months. At a median 6 months of follow-up, no responders in the trial progressed. Tumor response was assessed by tumor volume score, which was 56% with pexidartinib and 0% with placebo. Responses also correlated with improved patient symptoms and function. TGCT is a non-malignant tumor of the joint or tendon sheath. The disease can be locally aggressive and debilitating and is associated with severe morbidity or function limitations. In melanoma, the FDA granted a breakthrough therapy designation to the combination of bempegaldesleukin and nivolumab for the treatment of patients with previously untreated, unresectable, or metastatic disease. The designation is based on results from a cohort of the ongoing Phase 1/2 PIVOT-2 trial, in which the combination led to an overall response rate of 53% by independent radiology review, which included a 34% complete response rate in this patient population. Updated findings that were presented at the 2019 ASCO annual meeting showed a disease control rate of 74%. In PD-L1 positive, PD-L1 negative, and PD-L1 unknown tumors, the ORRs were 62%, 43%, and 33% respectively. There was an 100% reduction in target lesions in 42% of patients. Moreover, at a median 12.7 months of follow-up, 80% of patients had an ongoing response. The median duration of response was not reached, the median time to response was 2 months, and the median maximum reduction from baseline was negative 55%. The addition of atezolizumab to platinum-based chemotherapy significantly improved progression-free survival in patients with previously untreated, locally advanced, or metastatic urothelial carcinoma, according to results from the Phase 3 IMvigor130 study. In the Phase 3 IMvigor130 study, 1,213 patients with locally advanced or metastatic urothelial carcinoma who had not received prior treatment for advanced or metastatic disease were enrolled. Patients were randomized to receive atezolizumab alone, atezolizumab combined with platinum-based chemotherapy of gemcitabine with either cisplatin or carboplatin, or the same chemotherapy option combined with placebo. Additionally, reports showed that a positive trend in the co-primary endpoint of overall survival was noted at the interim analysis, but the data are not yet mature. No new safety signals were reported with the atezolizumab combination as compared with historical data. The IMvigor130 data are expected to be presented at an upcoming medical conference. In non-small cell lung cancer, treatment with afatinib followed by osimertinib led to a median overall survival of 41.3 months and a 2-year OS rate of 80% in patients with EGFR T790M positive disease in a real-world setting, as seen in updated interim results of the Phase 3 GEOTAG study. Moreover, in patients with exon 19 deletion positive tumors, the median OS was 45.7 months and the 2-year OS rate was 82%. The median time on treatment for sequential afatinib and osimertinib was 28.1 months and 30.6 months in those with exon 19 deletion. Following treatment with afatinib, the median time on osimertinib was 15.6 months overall and 16.4 months for those with exon 19 deletion. The updated median time to treatment failure with afatinib and osimertinib was 28.1 months. In patients with exon 19 deletion positive tumors, the median TTF was 30.6 months. A final analysis is planned for early 2020 and will include updated data from Asian and European countries. Results of an analysis showed that cell-free DNA-based detection of microsatellite instability status was found to be highly concordant with tissue-based MSI testing. Findings showed that incorporating MSI detection into the 74-gene panel liquid biopsy assay Guardant 360 demonstrated high concordance in approximately 1,145 cell-free DNA samples for which MSI status based on standard tissue testing was available. Cell-free DNA MSI evaluation was linked with high specificity, precision, and sensitivity with a limit of detection of 0.1% tumor content. In 949 unique evaluable patients, cell-free DNA testing accurately detected 87% of patients reported as having MSI-high tumors, and 99.5% of those having microsatellite stable disease, leading to an overall accuracy rate of 98.4%. The positive predictive value was 95%. This week, we sat down with Dr. D. Ross Kamich of University of Colorado Cancer Center to discuss targeting BRAF in non-small cell lung cancer.
Advanced non-small cell lung cancer is still a very serious disease, but for certain subgroups of patients, we are seeing long-term control of the disease. So we're moving from this binary choice. You know, you're alive and cured, or you're dead. And there is a middle ground where you are living in harmony with your cancer, and it is controlled in the same way like some other chronic diseases like diabetes or asthma might be. And for example, with ALK-positive lung cancer, we now have people living, at least the recent series from our center, nearly 7 years as a median. That means many people are living longer than that. In terms of the therapies for non-small cell lung cancer, we really can split it into two broad categories. So we have the standard ones which are going to apply to pretty much everybody. So some kinds of chemotherapy, some kinds of immunotherapy. And then we have individual pathways for defined molecular subtypes of lung cancer. There are now 5 FDA-approved molecular-specific subtypes with approved targeted therapies. That's EGFR, ALK, ROS, BRAF V600E, and NTRK gene rearrangements. But there's also a range of other molecular abnormalities that there are drugs, certainly in trials, which are looking very active, and no doubt these are looking for FDA licenses, or you could send patients for clinical trials, and that would include MET exon 14 skip mutations, high-level MET amplification, HER2 mutations, EGFR exon 20 insertions, to name a few. BRAF mutations are most well described in melanoma, but they also occur in lung cancer, particularly BRAF V600E, which is about 50% of the mutations that it's about 50% of the BRAF mutations in lung cancer, and only about 2% of lung cancer has BRAF mutations. The other ones are a range of different mutations, most of which we don't know what to do. So they're traditionally called non-V600E mutations. We saw some data at ASCO 2019, maybe some provocative cell line data. They might respond to MEK inhibition, but I think they're very much a work in progress. What we do know, and we know what to do, is when you have one of those V600E mutations, and as I said, that's about 2% of lung cancer. BRAF V600E mutation is a classic abnormality. It locks a kinase in an active conformation, and signaling through BRAF, it goes RAS, RAF, MEK, ERK, and so it's part of the proliferation pathway. And so the goal is to inhibit it. And the initial drugs were BRAF inhibitors, and they certainly can work, but they have about a 30% response rate. What it turned out was that there was a feedback pathway which limited their activity, and you could block that if you also gave a MEK inhibitor in combination. So what you now find is the licensed treatment in lung cancer as of 2017 is a combination of dabrafenib, which is a BRAF inhibitor, and trametinib, which is a MEK inhibitor.
That's all for today. Thank you for watching OncoLive News Network. I'm Kristi Rosa.